Hi, my name is Wenyu. I'm a PhD student at the University of Helsinki. Today, I'm going to talk about the strategy that our team, NETPHAR, used in the drug target prediction challenge. Before that, I would like to first talk about our motivations in participating in this challenge. Our group has been working on preclinical studies of cancer drugs by applying informatics and computational approaches on various types of perturbational data. Drug mode of action is one of our main focus, as it is the basis for both developing new drugs as well as repurposing the existing drugs. However, a recent study published in Science Translational Medicine showed that the drugs in the clinical trials are killing cancer cells independent of their reported mode of action. This further motivated us to reinvestigate drug activities in a more systematic manner. CTD Squared Center provides this challenge as a very unique chance to explore the computational potentials for investigating the drug mechanism of action in an unbiased manner. The organizers have also generously provided not only the viability result, but also gene expression changes for the same set of drug cell pairs. And both two types of data are generated under the same research center, making it a very valuable resource for pharmacological modeling. When we were given this data in the beginning of the challenge, we realized there are three key aspects in our task. So first, with viability data and expression data, how do we construct features for modeling? Such construction is also associated with the way we make use of data from public resources. And then once we have the data, how do we make the final predictions? First, gene signatures, or drug perturbational gene expression data. It is basically a high-dimensional characterization of drugs. What we are given are cell-specific signatures that capture the variations in cell lines. However, there is very little data point available from public resources. So instead, we use the consensus signature data, which behave very similar to chemical fingerprint and has been summarized over different cells, dose, and perturbation time. Hopefully, these consensus signatures can capture the core transcriptomic impact of each drug. When it comes to viability data, we need to decide which sensitivity metric should we use to summarize our dose response data. Metrics such as IC20 or IC50 only represent the drug potency and may fail to differentiate drugs' behavior under certain situations. In comparison, area under the dose response curve or AUC-based relative inhibition can represent both drug efficacy and potency. And that should be able to provide additional information during the modeling. Indeed, in our post-challenge explorations, we found that modeling with relative inhibition has provided significant better result than using the IC20 alone. So to derive the public available drug sensitivities, we used RECOMB, a one-stop database for both single drugs and combination drug screening. The database provides both response data and the fitted sensitivities by using various types of sensitivity matrix. So for drug target information, we use the DTC, Drug Target Commons, which is published in 2018. DTC is a community effort for curating and standardizing the drug target interactions from various experimental resources. For each drug target pairs, the DTC use this microball assay ontology to harmonize the heterogeneous results from different detection technology, assay formats, and endpoint mode of action into one unique score, which is on the 0 to 1 scale. This kind of score fits well to the current project. So in the end, what we have is a set of 973 gene signature features, as well as drug sensitivities in the 11 cell lines. Our implementation include first, calculate this pairwise similarity using this data, and then use this pairwise similarity matrix together with the drug target information to make the final predictions. Our prediction method is basically adopting this common two-step strategy. So first, we transform the data point from the original data feature space to the similarity or distance space. And then from the similarity or distance space, we form a model to do the final prediction. We believe such a framework helps the detection of signal from inherently noisy dataset with small sample size. And this strategy is actually used by quite many methods, from classic k-nearest neighbors to a full kernel models. And with varying flexibility in transformation steps and also prediction steps. Currently, our group members are exploring if a more flexible kernel function combined with penalized regression can provide even better prediction result. To conclude my talk, I have 3 take-home messages for you. So first, drug targets can be predicted from drug responses and perturbation of profiles with proper modeling. In addition, we found relative inhibition is a robust estimate of drug response that can be used in such kind of predictive modeling. More importantly, we found deeply curated pharmacological databases such as DREAM and Drug Target Commons is the effort of data cleaning and pave ways for computational projects just like this. We would like to thank you, the organizer, for this excellent challenge. And we appreciate the support we got from our group members, as well as the computational resources from CSA IT Center in Finland. Our effort has been funded by the European Research Council, as well as Academy of Finland Research Fellow Grant. That's all for my today's presentation. Thank you for your concentration behind the screens.